Case Western Reserve University's Institute for the Science of Origins proudly presents the Origin Science Scholars Program. The Institute advances the scientific understanding and application of origins and evolution of human and natural systems. The Origin Science Scholars Lectures are presented with the assistance of Case Western Reserve University's Siegel Lifelong Learning Program, College of Arts and Sciences, and Media Vision. Tonight, it's my pleasure to introduce our speaker, Patricia Princehouse, who is a fellow of the Institute for the Science of Origin, but also its associate director. She also directs our Origin Science major and the university's program in evolutionary biology. A native of Ohio, Dr. Princehouse earned her PhD from Harvard University, where she worked with Stephen Jay Gould and Richard Lewontin. Her primary interest is in the evolution of apes. She was honored with the National Center for Science Education's Friend of Darwin Award for safeguarding the integrity of science education in Ohio's public schools. Tonight, she will be telling us about zombie DNA. Why do vestigial genes and organs persist? Please join me in welcoming her.
We're going to talk about zombie organs, zombie DNA, what it can tell us, and zombie mathematics. Are we 98.6 zombie chimpanzee? What does that mean? Why zombies? Well, as Glenn mentioned earlier, we have a game on campus, Zombies versus Humans, that happens every time this year, so it seemed appropriate with Halloween coming up. So anyway, what about zombies? They persist in an undead state. They're reanimated, but they're changed. They're different from how they were before. They supposedly eat brains. Well, you know, every analogy breaks down sooner or later. Although there have been zombie appendix attacks. My husband was in the hospital very badly for one. Seriously, potentially fatal. So it does happen that zombies attack. When we're thinking about similarities, right, analogies break down. After all, zombies are only a little bit like vestigial organs. But in biology, there's a special kind of sameness that does hold up. It's called homology. This is a picture from 1555, someone writing a treatise about birds. And it's very striking when you look at the skeletons together, right? Richard Owen coined the term homology, and he said it was the same organ in different animals under every variety of form and function. Now, people often say, and that was in 1843, before Darwin published, people often say that Owen coined this term in a non-evolutionary setting, but that's not really the case. It's confusing, especially for folks that don't know the history very, very intimately. And online at the Berkeley site, I found this quote trying to explain Owen's thinking. It says, in later years, he alternately denied its validity, professed ignorance on the matter, and claimed to have come up with the idea himself 10 years before Darwin. Right? Now, how can that happen? Well, obviously, there's more to Owen than the person writing this realized, because he's not a ridiculous character. He's a very smart man who was instrumental in establishing science as a profession in England in the 19th century. It's that he had an unpublished theory called the law of ordained continuous becoming. Okay, which is a kind of evolutionary theory. It doesn't have a mechanism like Darwin's. And so what he objected to in Darwin's was natural selection. He thought there was some natural way by which organisms changed into one another, but that this sort of red in tooth and claw Darwinian natural selection thing had to be wrong, right? He said that the divine mind planned the archetype. And so all of these homologies that you see are part of this archetype. And that he foreknew all of its modifications. So if you think of God as a being that exists outside time and space, of course he knew all of its changes that it would go through. He knows everything. So it's not really a problem that way. Owen did have this drawing. He called the vertebrate archetype, right? Thinking about all different animals. It was not intended as representing an ancestor, but and of course there were very few fossils at the time that were well known. And Owen did, by the way, coin the term dinosaur. Right? But anyway, if he had found fossils like these from the Cambrian, they look an awful lot like what he expected the archetype to be. So this interplay between similarity and ancestry is a close one. Here's another picture of this is the fossil form haiku ichthyes, or haiku fish, of which a lot have been found in China. And so at the bottom there is Owen's archetype, and in the middle is a reconstruction of the fish from a redrawing of the fish from the fossil sediment, which you can see at the top. And on the bottom right is an artist's rendition of what it might have looked like in life. So anyway, Darwin then took Owen's idea of homology and made it much more distinctly evolutionary descent-oriented. He says, the framework of bones being the same in the hand of a man, the wing of a bat, the fin of a porpoise, the leg of a horse, the same number of vertebrae forming the neck of the giraffe and of the elephant, and innumerable other such facts at once explain themselves on the theory of descent with slow and slight successive modifications. And here's pictures, you've seen ones like this, where the homologous bones are represented under every variation of form and function, right? And so under Darwin's view, you have these same skeletal structures. They take on different shapes due to different selection pressures. So they're adapted to different roles in ecology, right? So that explains why you get changes, why you have homologous bones, why natural selection changes things sort of for new purposes or new roles. But what about things that have no current utility? Why do those change? And why don't they go away altogether? Here's our friend the appendix again. Here's a rabbit intestine with the full cecum, which ours is completely reduced, and the appendix on the end. That's a very functional system. The rabbit eats food. It goes into the stomach, into the intestine. It ends up in the cecum, where it sits around for a while. Rabbits actually excrete the cecum contents, eat them again, and they cycle through that way. Cecum contents are not feces, but sometimes you'll hear people say that rabbits eat their feces. They don't. They eat only the cecum contents, but they do expel them the same way. Anyway, the appendix is at the end of that. That's a very functional system. We don't use that system anymore, and so we have this little appendix that hangs on. Well, if it's not being used for something, why doesn't it disappear altogether? Because it is an opportunity for infection, such as the one that my husband got. Similarly, goosebumps. We get goosebumps when we're cold or when we're upset about something, when we're scared. Right? These are exactly homologous to what causes a chimp's hair to stand up and bristle, except that it looks like nothing when we do it. Right? Our hair stands on end, no one's impressed. Right? Why do we still put all the energy into having a very good working system that makes these tiny little hairs stand up? Right? They're vestiges of evolutionary history, just like things that have been co-opted. Whales develop hind legs in utero that never see the light of day. And a pelvis that hangs around. Famously, horses over evolutionary time have lost digits and are now left with just one hoof. Right? And it's really the middle finger that they're running on. But you'll sometimes see horses born with extra hooves. Right? The program that creates the hoof hasn't left all of its baggage behind. If you have cells there, it's going to make a hoof out of them, just like the three-toed horses of the past. Right? Now, this one's not functional. It doesn't have the details of the system still. But you can see the paths of evolutionary history by looking at interesting oddities like this. Here's another one. This foal is just been born, and it has a really quite impressive second hoof there. Sometimes there'll even be two. Similarly, there are blind animals all over the world. These are blind cave fish and their sighted relatives at the top, a blind cricket, a blind crab, and a blind salamander that live in darkness in caves. Darwin was fascinated by these things. He says, in some of the crabs, the footstalk for the eye remains, though the eye is completely gone. The stand for the telescope is there, though the telescope with its glasses has been lost. So you can have sort of something that disappears but not entirely, or something where parts of it disappear and other parts seem to remain intact. So the losing of things is a really interesting thing. It now turns out that actually some of these things are co-opted, so that parts of the brain that had been given over to vision, within a few generations already start getting co-opted just physiologically for olfaction. And over evolutionary time, you'll get this recruitment of the brain, the parts of the brain that were being used to see in order to smell better, and that makes perfect sense as an adaptation, right? But Darwin didn't know about that. He said, it's difficult to imagine that eyes, though useless, could be in any way injurious to animals living in darkness. I attribute their loss solely to disuse, right? So you will hear this. This is an argument that has taken place over generations, really. What good is, why don't you just not have the, the organ, right? Why not just keep it intact, but not use it? Why do things shrink up, right? Why does the appendix get smaller? And so the adaptive explanations that have been given are things like, that it takes up energy to grow. And so the smaller it is, the better off you are. Or because it's a site for infection, things like that, that there would be selection. So looking for selective ways to eliminate them. Why is it so hard to actually eliminate them, which would seem safer in some of these instances? And the reason is that the developmental program that creates individual beings, right, is very tightly constrained. And so it works sort of from the bottom up. You have genes that turn on and off. And to eliminate something altogether from this system would actually take enormous energy. You would have to have just the right variations arise and have strong selection pressure for them. So it's actually much easier for the organism to keep them intact in some manner. It would be almost impossible for A, the variation to arise, and B, for selection to act on it in order to actually get rid of these things. And that's why you have these vestigial parts. And people say, oh, well, the appendix, it produces some, a few things that aid in digestion. Well, it's not going to turn off all of its functions just because it's no longer being used the way a rabbit uses it. So it remains and it works with the rest of the system. And so thinking about organisms as integrated wholes, and not just the adult forms, but also the development from zygote on up. You can see how these things that turn on and off at different times, the timing is extremely important, how they would be extremely disrupted in order just to remove this little appendix. Right? And so those are the sorts of considerations to think about for that. So with that, we can go to some questions.
Is there a time scale known for when cave salamanders, cave fish, other things lost their eyes?
It turns out that the blind versions are generally related to the sighted fishes that are local to that area. And so in some cases you'll have splits that go back millions and millions and millions of years. In other cases you'll have some that are really only a few thousand years old. And yet it has worked that rapidly.
You spoke about how difficult and how much energy would be required to eliminate an unused organ. In my strictly unscientific mind, I have a vision of an unused organ shriveling and losing size and becoming so compact that it practically disappears, even though there may be a dot of it left. Does that happen?
So if you think of the appendix in this way, you can think of it shriveling up that way, except that the innovation that Darwin's view had, especially as it has developed to how it's used today, right, the evolution by natural selection, the end product, the adult form of an organism, is not the one that gets a lot of selection pressure. It's sort of along the way. And so if it shrivels up during one's lifetime, that's not going to have any effect on progeny, right? And Darwin was not entirely convinced of that in his lifetime, but he was looking for what we call a hard inheritance model, one that would not rely on things that happen in the organism's lifetime. In this case, the explanation that he gives there, when he says disuse, right, he attributes it entirely to disuse. That is exactly what he's talking about. It gets used less and less every generation, and eventually it would go away altogether. And so in that view, the appendix is something that just hadn't quite had enough time to entirely disappear. He was unaware of the integrated nature of genetics. He would, I can assure you, love to know about these things if he were around today. But he couldn't, he didn't know it then. He didn't know how this worked. He didn't know that the cells that are going to become the sex cells, the ones that make the eggs and sperm, those are sequestered very early in development in vertebrates. So you have the zygote. It divides several times, and then the cells that are going to become the ovaries and testes are kind of set aside, and the rest of the organism grows around them. You can think, why does that happen? Well, one selective reason that's been suggested is that the fewer divisions it undergoes, the fewer mutations are going to be introduced into that line, right? And so those are the ones that give rise to the next generation. So in order for there to be feedback into those which are set aside early on and don't turn on until puberty, it has to be something that would prevent or promote the organism having more offspring. And so if you have a developmental system, which is enormously complex, that produces a larger or smaller appendix, depending on which is desirable, if your organism with those genes for a smaller appendix reaches adulthood, has lots of kids who then reach the next generation, those genes are going to be retained and passed on, right, in an integrated system where everything works. If appendicitis takes you out at age 10, you're not, if you have the larger genes associated with a larger appendix, you're not going to contribute those genes to the next generation. So it's a much more complicated way in order for those genes to get into the next generation and affect the anatomy of the organism.
Thank you for joining us. You have been watching Dr. Patricia Princehouse discussing anatomical homology, the common organs and biological structures shared across diverse organisms as a result of evolutionary history. For more information on the Origin Science Scholars Program, please visit the Institute's website at origins.case.edu. In the next part of the talk, Dr. Princehouse will discuss how homology can extend into the genes. Now, back to the talk.
I'm trying not to use a lot of terminology, but it's really hard. I want to give you 4 terms. One is chromosome and haplotype. We'll put those one together. A chromosome, as I think most of you know, is a stretch of DNA in our bodies, and we have famously 23 pairs of chromosomes, right? Different organisms have different numbers of chromosomes. Chimpanzees and gorillas have 24 pairs. 2 of their chromosomes have, in evolutionary history, fused into 1 chromosome, and that's why we have 1 fewer, is we still have all the stuff, but it's put together in a different package. Okay. So the haplotype is the arrangement of genes along that chromosome. Right, so you can think of full chromosomes or haplotypes as chunks of those chromosomes. A locus, when we think of a gene, if there's a gene, like I was spewing around the term that there could be a gene for a smaller appendix, something like that, right? We would have to think of that in terms of a specific place along the chromosome where there is DNA that eventually ends up as protein and gets incorporated into this developing system. So you can think of it as an address along the chromosome where there's a gene. Alleles, which you've probably also heard of, are different versions of the locus. So you can have different sort of flavors of ice cream at that address along the chromosome. Again, the analogies just keep coming. So you can think of the allele that is often associated with having blue eyes, right? And the allele associated with brown eyes. These are versions that occupy the same place on that chromosome. And you can't have more than one at that address. So you have to have either the allele associated with blue eyes or the allele associated with brown eyes, or there are so many different loci. Some of them have 132 different flavors that you can have at that locus. It's enormously complex. So this is how sometimes you'll have alleles floating around in a population. You can think of this in terms of disease alleles, right, that don't show up. You'll say, well, you know, this child has a gene-associated disease. Nobody in our family have these diseases on either side, but perhaps back 12 generations ago there was someone that had them. It's been floating around, and you just unfortunately happened to end up with this child that had 2 copies of it. Yeah. A gene family gets a little bit tricky, but what happens is, again, to make the analogies even more ridiculous, you have your locus, which is the address of your house where your alleles live along the chromosome. You clone that house, and you insert it the next block over, right, or just next door. So you then have a second spot where you can have all of those same alleles show up. And this can be really useful. It can give new functions. For example, if you have you see squirrels that are, there's black squirrels and brown squirrels and gray squirrels that we see around here, right? Those are 1 or 2 allele issues, right? If you get and they're pretty discrete, right? They're either black or they're brown or they're gray. But if you had a gene duplication event that gave you a gene family for that, you might be able to have nuances of color in between the brown and the black, in between the gray and the brown, that sort of thing. And so the more copies of this, the more cloned houses you have, the more alleles can play a role in producing pigment that can be put down into the hair. And you know, this might just be entertaining for us, but given a particular environment, it might be absolutely crucial. And Cynthia, in her presentation 2 weeks ago, talked about the mice living on the different substrates, living on the light sand versus the dark sand. It can make a big difference if you get an allele that shows up that can produce a nuance of color that might adapt you better, right? So those are gene families. And yeah, the other thing you want to say about this is homology comes into this as well. So not only organs are homologous, but the genes themselves are homologous. In cloning that house, right, in the copying process, that locus gets copied and 2 copies get inserted instead of just 1, right? That's homology. It comes from the same ancestral gene. And so here's an example of gene families. This is for globin. The hemoglobin family of genes is extremely important and had a lot of study. There are things called pseudogenes in it. Pseudogenes are examples of how the houses, the loci, have been copied. New forms have been inserted, right? They've taken on different roles, and at some point a mutation has turned them off. So you had a nuance of color in that squirrel, and then some mutation has turned that off. It's not crucial for your organism. It doesn't get selected for or against. It just floats along, and pretty soon you've got a population of squirrels that don't have that nuance. Doesn't matter. Natural selection doesn't care about it, right? But it's not that, again, that pseudogene that's taking up a little bit of space on your chromosome, it's not going to get scooped out and thrown out. That would be enormously difficult. And energy consuming. So it hangs around. And so what you have is the genome that's littered with pseudogenes. Right? They're homologies. They're remnants of, vestiges of past evolutionary history. Right? The interesting thing is that if you look at different lineages of animals, our lineage, gene duplication event in the gene family that produced something called theta globin, we have a pseudogene for theta globin, right? We don't produce theta globin. It's got a mutation in it. It doesn't get transcribed. But horses still use theta globin, but they've turned off other ones. And so you see this sort of patchwork of homology going on. Different things are useful in different lineages. I'm not going to say too much more about that because I know it gets enormously confusing, but it is a fundamentally important thing to know about our biology and our evolution. Similarly, if you want to think about the genes or the genome, like the collection of genes, you can think of them as these interacting modules, right? So the loci, the houses, do these different jobs. On these modules, you have these alleles that do slightly different versions of those jobs. As these act inside your body, right, you produce more and more complicated things, gene products out of them. You create a hierarchical or quasi-hierarchical structure. Now, how those jobs are done, I want to address briefly. It involves looking at the cell. And I had a kid tell me once, in the cell, there's the middle part and there's the goo. So we're not going to talk about all these cell structures in there, right? We've got the middle part, the nucleus, and the goo, the cytoplasm. The goo has a lot of really important stuff in it, like mitochondria, which are descended from endosymbionts that we've had a couple lectures on, right? So they're homologous to this bacterial cell, right? They have a common ancestor, but one of the descendants, the one that became mitochondria, became endosymbiotic in evolutionary time with other single-celled organisms, eventually creating the nucleus and all of these other cell organelles. They're homologous to things that are outside of vertebrates and everything else, right? Give nature long enough time, it does bizarre things, okay? To make another analogy, all right, what happens in the center of this, in the middle, right, is you've got the DNA. Not all organisms have DNA. You don't actually have to have DNA in order to be life. RNA is sufficient, the one that we use as sort of a messenger and to build things, right? But in organisms like us, we have a sort of a library that RNA uses. It's like a blueprint library. And what the RNA does is it takes the chunk of it that it's going to use, puts it on the copy machine, makes a copy, and then it sends it outside the nucleus to the ribosome. The ribosome is like the construction site. That looks at the blueprint that has come out, and it does the things it needs to do in order to make protein out of it. The protein then folds up into different shapes, which are then used as the building blocks to make everything in your body. Right, so the RNA makes protein. The DNA basically does nothing. It sits there like a blueprint on the shelf. So it's really more complicated than that. In the nucleus, the RNA takes part of the DNA, it chops up the copied pages into a bunch of pieces, sticks some of them back together, and sends it out to the construction site that then does a similar thing. We won't go into that, but again, you get these layers and layers of complexity. And you can start to see why trying to get something simply to disappear is not an element that goes well with that system. So that's the main work of the DNA, is to be copied and make protein and build your body and keep you healthy and this sort of thing. It interacts, it builds the phenotype that interacts with the rest of the world. The other function, the one that I'm talking a little more about here, is just of the DNA. In a body cell, in a regular cell that's built out of the proteins continuously that makes you heal and sort of grow and things like that, when the body cell divides, everything is duplicated and everything is identical, right? Nothing changes. But when the sex cells divide, when you have eggs and sperm that are created, not everything is identical. And again, as you may guess, it's a complex process by which things line up, they get duplicated, they get pulled into daughter cells that also have 2 copies of each chromosome. One of each came from parent organism. Those then get cut down again, and the cells that end up becoming eggs and sperm have only half of our chromosome pairs. So they have 23 chromosomes instead of 23 pairs of chromosomes, right? Those then, the egg cell recombines with the sperm cell to create the 23 pairs again in the offspring. And so this is the pictures of how they line up, they duplicate. You you'll notice that they start out red and blue, and that they now are sort of checkered. And we'll talk about that in a minute. Here's another picture of it, which I find a little bit more interesting because you can see that you end up with 4 at the end. You have one cell, it divides, then you get 2 more, you end up with 4 cells. All 4 of them in the case of males, in the case of sperm, are active. They are not in the case of eggs. So on the pink ones there, you can see that you end up with 3 at the end that are just tiny. Those don't become eggs. You get 1 egg because 1 cell has to keep all the goo because of all the important stuff that's in the cytoplasm. And so that's why you end up with 1 egg, whereas you end up with 4 sperm from the same process. Sperm are more easy to produce, partly because of the cytoplasm problem. Because they don't need to carry that with them. They just need enough to make sure they have enough energy to go the distance that they need to go. Over 100 million sperm are produced every day at an average male person. But only about 1 egg is produced every month in an average female person. That means in your lifetime, you produce about 500 billion sperm, or about 400 eggs. So that's another complexity that leads to a bunch of different sequelae, right? So if you start to think about what are the different potential offspring that could be produced from this system, right? Well, you've got 2 people with whatever that number is, however it's said, right? Anyway, potential sperm-egg combinations in their lifetimes, right? And so the potentially genetically different non-identical offspring are quite a few, but it's actually far higher than that number. And it's far higher because of that thing that I pointed out of the different colors, right? There's a phenomenon called crossing over that happens during sex cell production. Right? And so although you start out with 1 chromosome that you got from your mom and 1 from your dad, and they unite and then all of your cells have that is true. But during sex cell production, they duplicate. The 4 of them cross over. They actually exchange bits and produce 4 non-identical gametes, right? Eggs or sperm. So it's like taking 2 decks of cards, right, and cutting them into each other, right, just by cutting. But you get some differences, but you still get large stretches of that haplotype, of those stretches of chromosome, right? Big chunks of them. It produces a phenomenon that I mentioned before called linkage, right? So you don't shuffle them the way you do a deck of cards. Alternately, you cut the deck and then you shuffle them, and you get a lot of rearranging. Instead, you can shuffle these only by cutting the cards a couple times every generation. And so you leave vast tracts of your DNA intact, but you do get some changes. And in order for something to be selected out, you'd have to find, especially if it was in between 2 genes that were very important, you'd have to find just the right crossover events in order to delete that thing out, to select it out. And it's unlikely that you would have a selection force that strong to do it. So as far as the question, are you 1 in a million? What are the odds? I said they're very much higher. We have about, at least most people are saying these days, and the estimate has come down. People used to say we have about 100,000 gene loci. Right? Now people are saying, well, it's probably only about 25,000. That's still a lot of gene loci, right? Now our chromosomes are different sizes. They have different numbers of them on each one, but I'm going to ignore that and say, let's just figure that you've only got 1,000 loci per chromosome and that they're sort of randomly arranged along it. So the kinds of mechanisms that you need to do mathematically to do this is called taking the binomial coefficient of the number of loci with these, say, 2 crossover events per generation. That gives you this large number of 900,000. Across all 22 chromosomes, you raise that to the 22nd power, you get this number that's 9 times 10 with 131 zeros after it, times 2 sex chromosomes. Conservatively, this is 10 to the 132nd, right? Now you can say, well, but not all of those loci have alleles that vary, right? So maybe it's not as high as that, right? But even if you assume just 125 variable loci per chromosome, you end up with a number that's still much larger than the number of atoms in the universe, right? We've got, using that lower, more conservative estimate, something that's about 1 trillion times the number of atoms in the universe. So when people say, is there enough variation for evolution to work on? I think there's enough variation, right? And yet, it is contained in this enormously complex system that produces similar effects, right? We can recognize that we're not the same person, but we all have 2 eyes, 2 hands, right? The system is very conserved that this variation is in, so it's contained. And that's that hierarchical structure that I was talking about. So it's modular, it's hierarchical, it has a lot of variation, but the variation is constrained. These are the sorts of things that feed into it. Of course, realistically, no married couple is going to produce more than about 25 kids, right? However, according to the Guinness Book of World Records, the record is 69 children born to Fyodor and Valentina Vasilyev in Russia in the 18th century. That was, and it's all documented in the monastery at Nikolsk, 16 pairs of twins, 7 sets of triplets, and 4 sets of quadruplets. So this guy remarried and had 18 kids by the second wife. They were no doubt, I mean, these are small villages, these were serfs, they're probably related. They were probably sisters or something. Anyway, you can get a large number of offspring that are going to be quite different from each other, but nothing like the variation that is potential.
DNA does have a mechanism to repair itself when it gets damaged. Do these repair mechanisms also work on the pseudogenes?
So DNA has hotspots for mutations where they're more likely to happen, places where they're less likely to happen. Very often places where they're more likely to happen have more repair mechanisms around. It's not clear exactly how that whole system works and what stimulates it and that sort of thing. But if it worked too well, there would be no evolution. Right? And so given the number of mutations that happen, the number of variations that arise, we know it gets defeated. It gets defeated typically with 1 phenotypic mutation per generation. So each of you has 1 mutation that arose just in you that has some phenotypic effect, probably very minor, right, that is unique in the world. Right? So there are enough things like that. In the case of pseudogenes, if the DNA repair mechanism happens to hit the right spot and it gets repaired, everything goes on as if it hadn't happened, right? But if it doesn't get repaired, and it really doesn't matter to the organism, it's not sort of visible to selection. There's another locus making that exact same protein, and you don't need it. It just turns off. There's no reason to get rid of it. There's really less than 10% of our DNA is actually working genes. The rest of it is stuff in between, which is repeated segments, pseudogenes, things like, well, even the pseudogenes is counted in that 10%. So there's a lot of room for that. I guess we have room for one more question.
If I'm peeling a potato and I gouge out my skin, it fills in. But if I cut off my finger, it's gone. Why?
It has to do with the complexity of the part that's missing, right? So if in early embryonic process, some cells went missing, those would likely be filled in and your fingers would be fine if that's the part. In fact, they are programmed to lose cells because they start out as a webbed flipper and then you lose cells in between. But once these cells have diversified into what they're going to be, bone, ligament, flesh, right? You gouge a piece out, that's enough it can fill in. It often fills in a little bit badly, there's a scar or something, right? But if you take the whole thing off, you don't have the kind of cells that you need in order to regenerate that. Maybe, I know they're working a lot on stem cells, those are the kinds of cells you need. It's possible that someday we'll be able to regenerate a lot of that, or to grow the tip of your finger in a petri dish, replicate it, and sew it on. But right now, we aren't quite there. But yeah, it's because it's the adult is sort of in fact the adult right before it dies is sort of the ultimate end product of what happens out of this hierarchy of effects. But all that really matters is what condition your finger was in at the time you reproduced. After that, it doesn't make a whole lot of difference. And so again, to have the system keep some cells that would be able to regenerate like that late into life, wouldn't make much sense. It would have to be really strong selection for that. There are some organisms that have that can regenerate tails or toes or things like that, but not mammals.
We hope you've been enjoying the Origin Science Scholars Program with Dr. Patricia Princehouse. Dr. Princehouse is the Associate Director of the Institute for the Science of Origins and Director of its Origins major and the program in Evolutionary Biology. In the second part of our talk, we learned how the division of sperm and egg cells introduces enormous genetic variation. In our final segment, Dr. Princehouse will tell us how we can understand our degree of relatedness to our family, fellow humans, and other species. Now, back to our talk.
This was just an example I was going to give of ABO blood groups, right? This is a gene family. These are alleles at the same locus. It's a polymorphism that has been around for many, many generations in primates, tens of millions of years. So there we are. I was pretty much done. All right, so what about this chimpanzee, right? What does it mean to be, whatever it is, 99 to 44 100% pure chimpanzee? You hear all of this in the public domain, right? And more specifically, how can I be 98% related to a chimp, 85% related to another so-called race, and only 50% related to my brother, right? Well, I mean, you haven't met my brother, but, or these guys. But anyway, both you and your brother get half your genes from your mother and half from your father. That is the case. On average, you share half your genes with each full brother or sister, 25% with each half sibling or cousin, right? We're talking about the stretches of DNA in this case, right? Not just loci or alleles, but just the actual stretches of DNA, chunks of chromosome, right? My friends, 23andMe, came back. It didn't say Scandinavia, but her brothers did, right? So what does that mean? Is that surprising? Is there something going on? Well, no, it's normal enough. She didn't get all of the stretches of DNA that her brother got, right? She got half of those, right? And she got the other half from her other parent. They got different things from each parent. So one parent is part Scandinavian. She just didn't happen to get those mutations that are known to have originated in Scandinavia. They were on the chunk of chromosome that she didn't get, but her brother did, right? So that means she did have ancestors living in Scandinavia, right? She and her brother share those ancestors. They were there. And that mutation arose while they were living there, or they would not have inherited it, right? But she just didn't happen to get that down the line, right? So the 50% number, again, has to do with the stretch of DNA, the specific ones that she got, not the whole history, not the whole story of one's ancestry. So all of the people in the photograph matter, not just her and her brothers, but all of them, if you want to get the full story. And going out from there, right, if you're going to get a full story of the ancestry of the human race, you want to include everybody, right? You want to know what happened in this coming to be. And this is a picture that Isaiah showed us last week. And you can see that most of the evolution of humans occurred in Africa over a very long stretch of time. And there are just a few of us whose ancestors left Africa. But there are some things that happened subsequent to that kind of stand out to at least those of us who are not from Africa, like skin color. And as was discussed last week, what we commonly call race is basically just skin color. There's really not much more to it at all. As Cynthia told us a couple weeks ago, skin color is related to the latitude that people come from. If they're in Africa, they need to have dark skin in order to block the UV light. If you're up in the northern or far southern areas, you don't need that because there's not as much UV light. But you do need sunlight to get into your skin in order to produce vitamin D. And so there would be selection for lighter skin in areas of extreme latitude, right? So having light skin doesn't indicate relatedness. There's another story of a friend of mine who had her 23andMe run. She's of British extraction. She's quite pale-skinned, right? Her DNA shows she's related to Cheddar Man, which is a 10,000-year-old skeleton that was found in England over 100 years ago, right? Recently, Cheddar Man's DNA was sequenced, right, to reveal that Britons still had dark skin less than 10,000 years ago. That was kind of a surprise to a lot of people. And when you look at it, you might expect Cheddar Man might be related to this Ivorian soccer star, right? There's some similarities. When in fact, he's far more closely related to my friend, dozens of thousands of years more closely related to my friend than to that guy. Studies consistently show that 85% of all human variation occurs within human populations around the globe, and that only less than 7% could be found within any one large racial group. Right? This led my advisor, Stephen Jay Gould, to say, if, God forbid, the nuclear holocaust happens and the only survivors are the Amatzosa people of the tip of southern Africa, 85% of all human genetic variation will have been preserved. So, if you're worried about that, don't be. The variation in the human species has been building for 200,000 years, really much longer than that if you think about ABO blood groups that go back, tens of millions of years. But what we perceive to be racial differences arose very recently, most within the past 20,000 years, and that race is effectively less than skin deep, basically. Light pigmentation, as we mentioned, is found in different populations at high latitude. The genes involved are different. These are variations that arose and were selected in those specific populations. There's not one, small set of white genes. They're different ones. So anyway, what about that chimpanzee? You can think about the 50% as the chromosome chunks that you share with your immediate family. You can think of the 85% as the alleles, the different flavors of the loci that you share with all other living humans, right? And you can think of the 99% chimp measurement as referring especially to the loci and to the gene families, right? So when you say we share 99% of our DNA, we share 99% of our loci, but they have, and some alleles, a lot of alleles, but the differences are some alleles, but a lot of different loci, so that in our lineage, we've had gene families develop that have been selected to do different things, right? And chimps have also had that going on in their lineage. So since the split with chimps, we've developed in one direction, including evolving new loci, new gene duplications, new gene families, and they have evolved in a different direction, right? But all of the stuff that we inherited from, over the past 4 billion years, is still contained in our DNA. Right? It's just that it has had these slight variations that have arisen over the past less than 10 million years that result in big changes, because you can tell your relatives from the apes. So who knows where our zombie DNA is going to take our species next? I don't know.
The Origin Science Scholars Lectures are presented by Case Western Reserve University's Institute for the Science of Origins, with the assistance of the Siegel Lifelong Learning Program, the College of Arts and Sciences, and MediaVision. For more information on the Origin Science Scholars Program, including a full video archive, please visit the Institute's website at origins.case.edu.